Hi, my name is Amy Burgin, and I am excited to share with you today an overview of a relatively new project that I'm building along with a great group of collaborators and co-authors. The project is the aquatic intermittency effects of microbiomes on streams. Like many in this special session, we are studying streams that switch from dry to wet conditions, sometimes in very dramatic fashion, as is shown here for one of our field sites at Kings Creek and Konza Prairie. So Kings Creek is one of the more well-studied intermittent streams because that's a feature of its hydrology. It has a very typical pattern of wetting up in the spring and drying out, and that's been characterized for 40 years by a USGS gauge that is there. And while we know a lot about some streams like Kings, we know very little about the much more abundant and prevalent intermittent streams that exist in every region of the lower 48. Of course, they're very abundant in places like the Central Plains where Kansas is, but they're also found in every other state of the continental US. And so AIMS was funded under a mechanism aimed at increasing collaboration and capacity in areas of high national need, with the theme of that program being harnessing big data to solve problems of national importance. And the challenge that we articulated is that despite their prevalence, we struggle to predict how intermittency affects downstream water quality due to a lack of data and physical infrastructure, gauges to help us understand them, methods for understanding the aquatic and terrestrial interface, and training in team science and big data tools. And this is an area of national importance because of its relevance for the waters of the US, or WOTUS rule, to the Clean Water Act. This was replaced in 2020 by the Navigable Waters Protection Rule, and that ruled back protections for intermittent streams and ephemeral streams. And this is now tied up in court involving several states, tribes, and environmental groups. And so AIMS's goals are clarified into three areas: research, education, and workforce development goals. Our research goal is to create a network of field sites integrating hydrology, biogeochemistry, and microbiome work to better understand these systems. Our education goal is to train scientists in collaborative, team-based open science approaches. And then the third goal is to provide workforce training on integrating these data sources like microbiome sequencing data, real-time sensor data, and GIS data. And I'll speak more to that. Ultimately, we not only want to train scientists to think about these systems, but we want to also make it more accessible for people who are already trained to start engaging in research in intermittent systems. And so that brings me to perhaps the most important part of my talk, which is what AIMS can do for you if you're interested in studying intermittent streams. We're embracing an open science approach where we're making all of our project policies, things like how we're doing mentoring, authorship, and data sharing and conflict resolution. We're making that all very public and open. We are also making our scientific protocols, standard operating procedures, open and available on something called the Open Science Framework wiki, where you can go and download any of the SOPs that you might hear me mention today. We also have a YouTube channel where we're sharing videos of how we operate these SOPs, so you can check that out as well. We are training people not just on our project, but people just interested in building their skills on being able to use these data sources through a mechanism called the Carpentries, largely operated in this particular way out of the KU libraries. You don't have to be at KU to take these workshops, but members from different organizations can sign up for them because they are open. And recently we've offered and helped with some workshops called such as R for spatial analysis, open science workflow for our studio, things like that, where people can come and get a training in a day or two to start building these skill sets. We're also developing code and functions for working with very common sensors and intermittency sticks, which I'll mention a little bit more here. There's a GitHub repo with some of the functions already posted to it that are going to be converted into an R package to be able to work with some of these kinds of data sets. And then finally, we have a lot of data and we want to find people to help us understand it. So if anything I mentioned in the latter half of this talk sparks your interest in terms of thinking about it from a certain angle or bringing a new lens to it, we would love to talk to people that are interested in writing pre-doctoral or postdoctoral fellowships. The postdocs tend to come due in the fall through either the Earth Sciences or through the Biodirectorate at NSF. We're also interested in talking to early career researchers who are in EPSCoR states and want to leverage a mechanism called a Track 4. I'd be happy to chat more about how we can do that. And if none of these apply to you, we're still interested in talking to people who just want to learn more about AIMS or potentially connect with some of the data that we're generating. We have a mechanism called an AIMS affiliate or early career researchers who want to leverage some of the research infrastructure that we're building to help build their own interest in intermittent streams. And so let me now switch gears to talking a little bit about what AIMS is and what exactly we're collecting and why. So as part of the AIMS project, we're predicting that intermittency, as defined as differential patterns of dry down and wet up, will manifest distinct hydrologic mechanisms across our 3 different regions. And with these distinct mechanisms, combined with regional scale climatic variation, we think those factors will govern water quality and nutrient loads via microbial processes. And so we're organized into 3 regions: the Western Mountains, the Great Plains, and the Southeast Forests. And our goal is to build a network of well-instrumented intermittent stream sites to start testing some of our hypotheses, as well as to continue doing work on intermittent streams. And that continuation is important because we have such little infrastructure to study intermittent streams in the US. And so these are 5 of the states that we have AIMS members in. And as you can see, in all of these states, about 50% to 80% of the stream miles are classified as intermittent. Despite that, very few, if any, gauges from the USGS are on these, are classified as intermittent. And that tracks with the national average, which is about 5.8% of USGS gauges that are classified as intermittent. And even fewer USGS gauges that are classified as intermittent also contain any kind of biogeochemical data alongside that hydrologic data. Only 0.04% are classified as having intermittent flow, as well as having a dissolved oxygen sensor. So there's really limited data on these national networks that already exist, and we're hoping to help fill some of that. And so this is the big picture perspective that I'll break down in a lot more detail about how we're studying intermittent streams and AIMS, arranged along temporal intensity and spatial intensity. And as I mentioned in the last one, the first tool that we're really bringing to this is using a lot of different kinds of sensors to understand where water is in the network and how that impacts water quality. A main sensor that we use are these sticks, stream temperature, intermittency, and conductivity sensors. Very simple sensors that are relatively cheap, and we can put out many in a watershed. There are about 50 dots here in this representation of the Kanza watershed, and each one of those represents a stick. And as you can see in the difference between the spring period and the wet period, the switching of the sites between being a color versus being black shows how much of that network dried down in a short period of time. And at the outflow of these, we are also using high frequency biogeochemistry sensors, which I'll describe in a lot more detail, to understand how different parts of the watershed are turning on and off and how that's affecting what's coming out of the watershed. The second approach that we'll talk about is using distributed seasonal sampling, going out to key areas of the watershed at 3 times a year and trying to do integrated sampling to understand how there might be some seasonal component to intermittency. The third approach is a synoptic, a very intense synoptic campaign where we go out to 50 locations within a site and take a very intense one-time snapshot of where there is water in the system and how that is affecting the microbiome and biogeochemistry. And then the fourth approach is an experimental approach where we're going to experimentally dry down a section of stream and try to understand how that affects the microbiome and the biogeochemistry. And so this arrays along an axis of increasing emphasis on process and increasing emphasis on context that allows us to understand our region specifically, but also hopefully some global drivers across regions. So a little bit more detail on each of those. Approach 1 uses these real-time sensors, as I mentioned, these sticks that are arrayed out. In general, we have 20 to 90 per watershed in 9 different watersheds, 3 watersheds per region. This is a map of the stick wetness in June that was made by Chris Wheeler. These sticks have been out for about a year now, so this map at end of a year represents about 1 million data observations about where water is on the watershed. So here's how the sticks have dried out just between June and August, as I mentioned on the previous slide. Within this watershed, we've identified 7 spots we call long-term monitoring sites where we've also put in groundwater wells. And that allows us to get at groundwater chemistry and connect it to the surface water chemistry, understanding that connection between surface and subsurface. And then finally, we have our real-time sensors at the outlet that allow us to get at water level, as well as temperature, conductivity, dissolved oxygen, nitrate, FDOM, and turbidity, as well as in one site in each region having a SCAN, multispectral data in 10-minute increments. So this allows us a really intensive look at what's happening in the watershed when certain parts of it turn on and off and how that affects downstream water quality. The second approach is using distributed seasonal sampling, where we take a lot of different chemistry, microbiome types of samples, macroinvertebrates, and hydrology, or at 3 different time points at those 7 long-term monitoring sites in 3 different seasons per year in 2 different years. So this allows us to have a data set that's very highly coordinated and comparable, executed the same way across all of our regions, to start to compare different drivers of intermittency and its effects on the microbiomes and the biogeochemistry. The third approach is a spatially dense synoptic campaign. So we did this in June of 2021 at Kanza, where we went out to all 50 stick locations and collected that entire suite of things that I mentioned for the approach 2. So we basically marched from the bottom of the watershed up to the tippy top, where it's barely where the stream is born. Of that, we had about 46 sites that had water samples, and all 50 sites we collected microbiome samples. That was many freezers full of samples in a very short time period. It took a small army of people, many of whom were really excited to be out doing this kind of coordinated campaign effort after we had a lot of lockdown issues from the pandemic. We managed to collect a lot of samples in a very short amount of time. We'll repeat this in Alabama in just a few weeks, and we'll also do it in Idaho in the summer of 2023. The fourth approach is an experimental stream dry down, wherein we will take a stream and apply a Backy experimental design, where for 2 weeks before, we'll have a control section and an impact section. We will monitor the dissolved oxygen, the water chemistry, the macroinvertebrates, and the microbiome, kind of centered on our main sensor array at the outflow of a system. And then we are going to, or have already applied a dam structure. This is the brainchild of Carla Atkinson and John Instead. So on the end of each of these pipes, we will, each of these tubes, we will attach a pipe, and that will kind of short circuit the stream, allowing the water to pass over it, drying out the channel, which will happen for 4 weeks, during which we'll collect everything I mentioned above, as well as do some mapping and decomposition experiments to understand how different microbial processes are affected by the dry down. And then we'll re-wet it after 2 weeks. Again, we're doing everything above, but doing more temporally intensive sampling to understand how the re-wetting event affects the water chemistry and the microbiome samples. So the take-home messages that I have for you today are that AIMS is a highly coordinated, integrated data collection that will allow us to test hypotheses connecting intermittency to downstream water quality across 3 really different regions. And the infrastructure that we've invested in and put a lot of energy into building expands the number of sites that we can work at and the types of things that we can do on intermittent streams. We are using a really open science approach. So take our stuff. If I've mentioned something and you can't find it on one of these resources, there's QR codes down here to find the resources I mentioned. If you can't find something, please reach out to us, and we are very happy to share and collaborate with others. And finally, we have a lot of data, and we would love to find more people that want to study intermittent streams with us and the data that we're generating. So if you're interested, please reach out to me or one of the other team members. And the other team members will also be presenting work at JASM on Monday. We have a number of posters that will be presenting, many on the Kanza Synoptic from last year. And then we also are co-sponsoring a special session on Friday afternoon, where 5 different people will be talking about different aspects of AIMS and sharing a little bit more data than I was able to get into in a short talk. So I would like to also acknowledge the many people who are not co-authors, but still contributed importantly to this effort. And especially those that are allowing us access to these sites so that we can work on these intermittent systems. And I appreciate your attention to this talk. Thank you.